Welcome to Unit 2. We'll begin our discussion with concepts in Chapter 3 regarding the patterns of inheritance of genetic traits in man. So we'll begin with monogenic or really single gene traits. These are also known as Mendelian traits because the father of genetics, Gregor Mendel, was the first to categorize these particular types of traits. So again, it only involves a single gene. Now, right now, we know of more than 19,000 single gene or monogenic traits that have been defined in humans. The majority of them are on the autosomes with more than 18,000. A little bit less than 1,000 on the X, and I'm not sure whether that 57 is still correct. There might be a little bit more on the Y chromosome. Now, when Gregor Mendel studied the genetic traits, he studied just 7 traits in the common garden pea, but he found 2 central principles. Now, this isn't in your textbook, but you can find it under other sources, and I just thought it was important enough for you to get a handle on what he discovered just working with the simple garden pea. The first principle was, of course, of segregation. And I'll put a little definition here in quotes. The principle of segregation states that sexually reproducing organisms possess genes that occur in pairs, and that only one member of this pair is transmitted to the offspring, i.e., it segregates. This means that genes remain intact and distinct, so they don't become blended when you inherit them. And that's a very important concept. For example, an allele for a round seed shape can be transmitted to offspring in the next generation, which can then be transmitted to its offspring, and so on. The other principle that Gregor Mendel noticed was the principle of independent assortment. This states that genes at different loci, loci, of course, we've already mentioned, is the plural for locus, and locus is talking about location. So at different locations are transmitted independently. So consider 2 different loci, each with 2 alleles. Pea height, short or tall, and seed shape, round or wrinkled. The allele transmitted at one locus, such as the pea height, short and tall, will have no effect on which allele is transmitted at the other locus, the seed shape, round or wrinkled. And that's what he was talking about. So this is that genes that are on different chromosomes or on the same chromosome but far apart are transmitted independently. All right, getting back to your textbook, we'll start with a course of definition of phenotype. And this is the actual physical or clinical expression of an individual's genotype. In other words, what you see or the diseases that they develop. Now, if you're talking about individuals with 2 different genotypes but have the same phenotype, chances are you're talking about a dominant disorder. They're both homozygotes and heterozygotes, therefore are affected. But if you have the same genotype but may produce different phenotypes in different environments, then we're probably talking about a recessive disorder. And one of the most common ones are phenylketonuria, and we'll be talking about that a little bit more in a few slides. So you should be aware of the fact that phenotype results not only from your genotype, but interaction with the environment as well. So let's talk about some patterns of inheritance, and we'll start off with autosomal recessive inheritance. This means that because it's recessive, it's fairly rare in a given population. However, the heterozygous carriers are going to be more common than the affected homozygotes. Parents of affected children will usually both be heterozygous carriers. And the Punnett square that I've now put over there shows that 1/4 will be normal homozygotes. Half of them will be normal heterozygotes, and of course, fortunately, only 1/4 will be the affected homozygotes. The characteristics of autosomal recessive inheritance: one is observed in one or more siblings, but not necessarily or not in earlier generations. On average, only a quarter or 1 in 4 of the offspring of two heterozygote carriers will be affected. Males and females are affected equally because they're on autosomes. Consanguinity, in other words, inbreeding, is observed more often in pedigrees with autosomal recessive diseases. So we see these in isolated populations in very rural areas. We see this with such as the Amish in Pennsylvania, or some small towns in Canada or Maine. This is not so uncommon. And then there's another one that is one that we see, tyrosine negative albinism. This is an enzyme required for the synthesis of melanin and also normal optic fibers. So if it's inactivated, there'll be little pigment in the skin, hair, and eyes, and they may even have eye disorders. So this is one mechanism for albinism. In this slide, we see an example of course, the albinism between two family members here, and also of pedigree, which I want to kind of go through. So if you take a look at probably grandparents, up here we have a heterozygote carrier and a homozygote normal. Now all of their children, of course, will be normal, except they may have some carriers, all right? Now, as long as their carriers are marrying homozygotes who are normal, there is no problem. But now that we get into the third generation here, we have some first cousins getting together. And as a result of this, we have two heterozygotes mating, and of course an increased incidence of affected children as a result. So what you should get used to is whenever there's a case of consanguinity in a pedigree, they do use a double bar to indicate that. And the squares are always males and the circles are always females. I'm not quite sure what that's supposed to indicate, if anything, but that's the way the pedigrees have always been. So what about the recurrence risk? Every time two heterozygotes who are mating, the recurrence risk, even if they already have one, is still going to be, an affected child, I should say, is still going to be 25%. If a carrier mates with someone who is a homozygous for the disease, however, then the recurrence risk is up to 50%. In fact, at this point, it will mimic the inheritance pattern of autosomal dominant. And we refer to this as quasi-dominant inheritance. And the only way you can really distinguish the two is through very large pedigrees. And that's why sometimes what you'll see when you go on to the Mendelian Inheritance in Man online, or OMIN, it should be Online Mendelian Inheritance in Man. But when you go in there, you will sometimes see the same disorder, and they'll list families where it seems to be an autosomal recessive, and others where it's an autosomal dominant, and it might just be a true autosomal recessive, but you've got the small family, so you've got a quasi-dominant inheritance, or in the case of part 7 here is you've got a heterozygote who married a homozygote who's affected. Anyway, other relatively common autosomal recessive disorders include phenylketonuria and cystic fibrosis. So let's talk a little bit about phenylketonuria, or PKU. Has a fairly common incidence for an autosomal recessive disorder, 1 in 10,000 Europeans, 1 in 90,000 Africans. The mutation is actually in an enzyme known as phenylalanine hydroxylase, or PAH. What happens is this is a very important enzyme in the breakdown of the amino acid phenylalanine, okay? And without it, when the people who don't have this eat a diet high in protein and everything, they get accumulation of a toxic intermediate or metabolite, if you will, and as a result of its accumulation, there's some severe problems. In fact, what it does is basically disrupt myelination and protein synthesis in the central nervous system. so they can be severe mental retardation. It also is going to affect production of dopamine and melanin, so they'll have problems with movement and pigmentation. So amazingly, just by restricting their diet, decreasing the amount of phenylalanine, but not completely eliminating it because they still need some for their proteins, they're fine. But they have to adhere to this diet their entire life. Anyway, elevated PHE during pregnancy in a developing fetus that has this can have severe effects. So it's important for them to check, especially if the couple has had an affected child in the past. So if we take a look, here's the actual amino acid, phenylalanine, and this is where the enzyme is supposed to come about and rearrange things. And as a result, of course, you have a problem. You don't develop a lot of this. And this is one of the, they can do a simple blood test to check for it for the screening. And here, of course, over here on the right, we see a classic pedigree with 2 heterozygotes coming and having 1 M4 affected children. Well, this leads us to looking at autosomal dominant inheritance patterns. These occur in 1 out of 200 individuals, but each individual disease is actually rather rare. So, obviously, the trait, because it's dominant, is going to be expressed in both homozygotes and heterozygotes. The affected offspring are usually heterozygotes. A normal parent will mate with an affected heterozygote parent. And so you can see that in this situation, since it's dominant, that 50% of the offspring will be affected. So AA is the normal parent. The other one, the capital A and little a, is the affected parent. And each child then has a 50-50 chance of getting the disorder. An example of this is postaxial polydactyly. It's simply the presence of an extra digit next to the 5th digit, but you can always impress everyone by saying postaxial polydactyly. I always like saying that for some reason. Anyway, it can be inherited as an autosomal dominant trait, and this pedigree that we see on the lower right shows that both sexes are affected equally, and both sexes are capable of transmitting it. So here is a heterozygous father and a normal homozygous mother, and of course we've got a father who is affected and marries another, a normal woman, but now they have affected women. So, and of course then now these ladies will be able to pass it on as well. So another thing that's important about autosomal dominant is to realize the traits are seen in each generation. And here's just where we get the extra digit. And here, of course, but this other picture, you actually see the bones, which is kind of cool. Another autosomal dominant trait is Marfan syndrome. It occurs in about 1 in 10,000 North Americans, so it's actually fairly common. It is truly a connective tissue disorder due to mutations in a gene called fibrillin-1, or fibrillin-1. For some reason, I never like to say that one. Anyway, it causes a number of different defects. For example, there's problem in the eyes. They are myopic, and they can have an ectopia lens. Then they'll have the classic skeletal defects, very long, slender limbs, a hollow chest, and arachnodactyly. Scoliosis in the spine is common, and joint hypermotility. And unfortunately, though, they can have some even more severe problems in the heart, mitral valve prolapse, which isn't all that bad, but you can get regurgitation. Aortic dilation and dissection, though, is pretty serious. Now, there are a lot of famous people that we suspect had Marfan's. Abraham Lincoln, as you see over here on the lower right, has always been suspected. I wasn't all that familiar with Sergei Rachmaninoff, but Niccolò Paganini, who was supposed to be the world's greatest violinist ever. I have read that he was either had Ehlers-Danlos or Marfan's because he had such hypermotility with the joints and such long slender fingers. He could play passages that nobody else can. And he wrote composed music for violinists that just about nobody can play. So Niccolo Paganini had something. We're not exactly sure whether though it was Marfan's or Ehlers-Danlos. And this gives you an idea. Obviously, we've got a nice little atom picture on our left, but you can see a German family here, all with the very long fingers, and you can see how that would benefit a violinist trying to get certain chords. There are things that he could do that I guess that no one else has been able to do since. Recurrence risk. You heard me use that term when we talked about autosomal recessive disorders. This recurrence risk is something that parents will ask you, and these are parents who have an affected child, want to have more children, and are concerned whether they will continue to have children with a particular disorder. So that's what they're concerned about. An occurrence risk. Now you're advising a couple that have no children but know that there is a disorder in the family and want to know what their probability of having an affected child will be. But you have to remember, and this is why I brought up Mendel's principles, is that each child is an independent event. So it doesn't make any difference if there are previous children. It will have no effect on the subsequent children. The probability will remain the same. All right. Now we can talk about X-linked inheritance. Now women have 2 X chromosomes, men have 1. So the question has always been, why didn't women have twice the lethal amount of X-encoded enzymes? Well, it's because, as I'm sure many of you are already aware, one of the X chromosomes in each of our cells has been inactivated. This, of course, compensates for the dosage, so now we have same level of enzymes and other proteins that are present on the X chromosome as men do. What you have to remember, though, is that the X inactivation in each individual cell is randomly determined, but once it's determined, it's permanent or fixed, if you will. All normal females, then, are actually mosaics, because some cells will have, say, the mother's X inactivated, and other cells will have the father's X inactivated. However, men are hemizygous because they only get 1 copy of the X chromosome, and obviously it has to be from mom. We can then talk about sex-limited and sex-influenced traits. Now, sex-limited traits only occur in one sex, and this is like anatomical differences. Sex-influenced traits are more common in one sex, but they can occur in both. Classic example of this is male pattern baldness, which is not X-linked. It's an autosomal dominant trait in males, but in females it functions as an autosomal recessive. The homozygotes have hair thinning rather than complete baldness like the men do. And unaffected heterozygous females will transmit the trait to their offspring. Now, what about sex-linked inheritance? Well, sex-linked inheritance occurs when genes are mutated and they're located either on the X or the Y chromosome. Some of the more common sex-linked inheritance disorders are obviously on the X chromosome, and this includes hemophilia A, Duchenne muscular dystrophy. There's lots of different muscular dystrophies, and some of them are sex-linked, and then red-green color blindness. They'll behave like an autosomal recessive trait in females because females have 2 X chromosomes. But, in other words, if they both of the X chromosomes that they get have the trait, then they're homozygous, and it doesn't matter which one gets inactivated because they both have it, they will express the trait. However, if they're heterozygous, then they will not express the trait. Remember, X inactivation is random, so the females are a mosaic, and in most cases, 50% of the gene product usually produces a normal phenotype. Males, however, they are hemizygous for the X chromosome, so if they inherit a recessive disease gene, that is on the X chromosome, then they get the disease. Characteristics of X-linked inheritance are as follows. Seen more in boys than girls. Trait cannot be passed from father to son because the disorder is on the X chromosome, and fathers can only pass the Y to the son. So these disorders are transmitted through carrier females. It may skip a generation as a result, and it can be passed from a suspected father to the daughters, who then will transmit it to half of their sons. And this gives you a picture of a pedigree. Here is a carrier. Whenever you see a dot inside a circle or a square, that means a carrier. And you can see that the boys would be affected. The girls are carriers. The girls are never affected. Now let's look at some common mating types. With this, typically what we see is that a carrier female has mated with a normal male, and so you can see that 50% of the daughters will be normal, and 50% over here, I should say, and 50% of them will be carriers. Meanwhile, the sons, 50% will be normal, and 50% will be affected. Other possible mating types, we have the mother that is normal and an affected father. So the mother is normal, the father has only that one X chromosome, and the disorder or mutation is on there. So all of the daughters will be carriers because that's all the father can pass on to a daughter is his X chromosome. All of the sons, however, will be normal. However, if you've got an affected father and a carrier mother, now you have a different situation. You have 50% of the daughters actually affected because there's two affected X. 50% of the daughters are carriers, 50% again of the sons are normal, and 50% are affected. Then there is something called manifesting heterozygotes. This is when a female inherits a single copy of an X-linked recessive disorder and they are affected. How can this happen? Well, X inactivation is random, right? So you expect more or less a 50-50 distribution. However, occasionally there's a slight probability that almost all of the active chromosomes carry the disease mutation, in which case they can then express the disorder. And so they are referred to as manifesting heterozygotes because they are heterozygotes. Usually they have a small proportion of cells with active normal allele, so whatever the disease is, it's typically less severe. So if it's hemophilia, they would be mild hemophiliacs. However, if this is someone who has Turner syndrome, now Turner syndrome are females who have a single X, no Y, then of course the disease can be just as severe as in males because they only have the one X chromosome. Well, since I brought it up, let's talk about hemophilia A, also known as classical hemophilia. Incidence is 1 in 10,000 males, 1 in I should say 100 million females. I was about to say a million, but that's 100 million females. Anyway, the disease results because the factor VIII, which is very important in the coagulation cascade, is either deficient or defective, one or the other. It cannot participate in clotting. As a result, what clot formation is formed, it takes it a lot longer, or it doesn't occur at all. This leads to severe bleeding, but usually it's delayed, any kind of wounds or bruising, and then of course it bleeds into joint, and this can lead to joint deformity. But the most serious situation, of course, is an intracranial bleed. Severity is going to depend on how much of factor VIII is actually produced. Less produced, more severe. Not too surprising. We have here some pictures of symptoms seen in classic hemophilia. And of course you can see the swelling and as a result of swelling into the joints in the knee. And of course this is all the bruising that you see. Now, remember when we learned about the different types of mutations last week? These can affect the disease severity. So, nonsense mutations, remember they lead to a stop codon, so not all of the protein is made. That is usually a severe form of the disease. A missense mutation, however, where all you're doing is exchanging one amino acid for another, and we get milder forms. Now, 45% of the severe cases, though, the factor VIII gene has completely been disrupted because there's been rearrangement or inversion, as they say, of the chromosome. So sequence has got inverted, and of course the gene actually got disrupted then. And then, of course, no protein is produced at all. But I couldn't resist, because I think most people are aware of the fact that hemophilia A is predominant in the royal families of Europe. And so we can go up here, and the first known carrier was Queen Victoria. And of course she married Prince Albert, and they're showing her direct descent from George III. Anyway, so we get to all the children that she had, which was quite a bit, and they were married to Duke of Hesse, the Germans. This is where all the Germans come in and everything in there. And here, of course, is Tsar Nicholas and Alexandria, and of course their son. And you could go through all this, because I'm not going to. But anyway, you can see the Spanish house, Russian, Prussian, British, and German. There wasn't anything until they married into Queen Victoria. Well, this picture, of course, is a picture of Tsar Nicholas II and his family, which, of course, were murdered during the Russian Revolution in 1917. And they finally found the remains and were able to identify them using the polymerase chain reaction and comparing the genes with members of the European royal families that are alive today. And now they do have actually a little memorial for them that you can go visit if you ever go to Russia. We can then talk about X-linked dominant inheritance. I think most people have heard of X-linked recessive, but not so much about X-linked dominant inheritance. These are fewer in number and prevalence, and as we mentioned some of them, you'll be kind of grateful that they are. Examples include hypophosphatemic rickets. In this situation, the kidneys can't resorb phosphate. Phosphate's kind of important. Anyway, the bones are not mineralized as well. That's what poor ossification means, poor ossification, and so they bend and distort. Another one is X-linked dominant is Continentia pigmenti type 1. So they've got abnormal skin pigmentation and teeth, obviously bladder problems, I think, and eye and neurological abnormalities probably leading to the bladder problems, and then only seen in heterozygous females. Rett syndrome, this is a situation where a particular gene, the MCP2 gene, is inappropriately expressed, and these people can have autism, retardation, seizures, and gait ataxia. So some of these can be quite nasty. What are the characteristics of X-linked dominant inheritance? Well, you obviously only need to inherit a single copy. Females are affected 2 to 1 as males because they have 2 X chromosomes, so they're more likely, to have it, unless, of course, it's lethal in males. No father-son transmission, obviously, because it's on the X chromosome. And all daughters of the affected father will inherit the disease gene and are affected because they have to get that X from their father or they won't get the female. And affected females are usually heterozygotes, and they have a 50% chance of passing the allele to daughters and sons. This is a pedigree showing this to you. So you see you have an affected female, okay, and then she will go on and pass it to another affected female. Remember, this is X Linked dominant, here we've got a male affected, again unaffected females and males. So this is another summary that you could read about it, but it's kind of an interesting, we don't think of X-linked disorders as being dominant. Unfortunately, there are not a lot of them either after what we just read about those that we know of.
Y-linked inheritance patterns are seen as well. The Y chromosome, however, has a lot fewer genes than the X chromosome. We've only identified a few dozen. For example, the initiation of male differentiation in utero, the testis-specific spermatogenesis factors, and there's even a protein expressed on the surfaces of male cells that women don't have that's known as HY, and it can sometimes lead to rejection of certain grafts if placed in a woman. However, there are several housekeeping genes located on the Y with homology on the X chromosome. And it turns out that what'll happen is even the X chromosome that's inactivated in women, those genes that are similar to the ones on the Y chromosome are not inactivated, so that we do have the same levels of those particular housekeeping genes as men do. So there are some parts, X inactivation is not complete. Anyway, Y-linked inheritance, obviously father to son transmission only. Now, I was trying to find one of these for the X chromosome as well, but for some reason I can't find one. But this has been around for many years with some of the things like, ability to remember and tell jokes over here, channel flipping, addiction to death and destruction movies. I like the T2. Mine, of course, was that I added inability to ask directions, therefore the lost gene, and total lack of recall for dates. Oops, I kind of like that. But anyway, so I tried to find one for the X chromosome and I couldn't find one. So ladies, I think we need to come up with some of our own. Anyway, here's a pedigree that will show you Y-linked inheritance pattern. Of course, it's just from men to men. It's pretty straightforward. Pseudo-dominant inheritance can occur. A classic example of this is hemochromatosis, and it's a parent-to-child transmission of disease that is typically an autosomal recessive disorder, but it will appear as though it's dominant, and this typically occurs when one parent is homozygous and the other is heterozygous. And as you can see with the pattern that we have over here on the right, it doesn't affect development in utero, but as they get older, they will develop it. And this leads to the hemochromatosis. Actually, this should be an O, not an A. Excessive absorption of iron, and this can lead to liver problems and a number of other things. And believe it or not, the mutant allele is present in 1 to 10 individuals of Celtic ancestry. So you've got a little Scotch or Irish in there, you may have a bit of a chance for that. Digenic inheritance can occur as well. It's a recently recognized form of genetic transmission. It occurs when an offspring, a son or daughter, inherit 2 different mutant alleles from their parents. They are heterozygous then for each mutant allele, and so they're referred to as double heterozygotes. Parents are normal, have 1 in 4 risk of having an affected child, just as they normally would. And the affected child will have a 1 in 4 chance of passing both alleles, because and as a result, I should say, the transmission appears as though it is a dominant disorder. Examples of this are retinitis pigmentosa and the Bardet-Biedl syndrome. And this gives you an idea of digenic inheritance. This didn't come out all that good in terms of clarity, but you can see that we're talking about different alleles, and it occurs when they get their different mutations, and if they're heterozygous, it's said for each, but now they have 2 different, so they unfortunately can have an interesting disorder that way. Another inheritance pattern is called mosaicism. There's actually two types. There's what we call germline mosaicism, when all or part of the parent's germ cells are affected by a disease mutation, so then they can pass it on to the children. And when more than two offspring have an autosomal dominant or X-linked disease in the family with no prior history, you have to start thinking about mosaicism. But somatic mosaicism occurs when the mutation occurs in one of the cells during a parent's embryonic development. So all the cells descended from that cell are affected, but the others are not. So some of the cells are normal, some of the cells are not normal. The parent, as a result, is going to have a normal phenotype, but it can again transmit it to multiple offspring. Examples of this include a very lethal perinatal form of osteogenesis imperfecta, which is not nice at all. In this figure, we see examples of somatic cell mosaicism as well as germline mosaics. And if we look over here on the left, we can see that at an early stage of the embryo, a mutation occurs in one of the embryonic cells. So the majority of the cells are actually going to be normal, but of course some cells will have the mutation. And so this is a somatic cell mosaic, whereas a germline mosaic, this occurs in the fertilized egg, and then so it's carried off, but not necessarily to all of the cells again, but it occurred in the fertilized egg. So it's just an earlier mutation. Another factor that can complicate figuring out inheritance patterns is something known as reduced penetrance. And it simply refers to individuals who have the disease-causing genotype. In other words, they have the mutated allele in their DNA, but they don't develop the disease, so they never get the phenotype, all right? They can be healthy, and however, they can transmit the disease gene to the next generation. A perfect example of this is retinoblastoma, which is a malignant eye disorder that usually occurs in children. It is an autosomal dominant disorder, but it has a reduced penetrance because unless they get the second mutation, they don't develop the tumor. So 10% of the carriers with this have an affected parent, and typically offspring do not have the disease. Thus, the penetrance rate is about 90%. So it's pretty high, but it's not 100%. And this is just an example of retinoblastoma so that you can see what it looks like in the child's eye. And then behind it, you can see that this is all a tumor over here on the left. And here's the penetrance. You can see an affected father in this case. And they go ahead and have another affected father. But otherwise, nobody else is too seriously affected. But here's our carrier. And of course, so you can see that it will not be 100% penetrance, but it's pretty high. Some disorders don't manifest until people get a lot older. So this is what we've referred to as age-dependent penetrance. And so there's usually a delay in the age of onset. This does occur in many diseases, and again is going to complicate interpreting the inheritance pattern of the disorder. The classical example of this is Huntington disease. It also used to be known as Huntington chorea. This is a rather nasty disease. Typically begins after 30 years of age or around 40. So they have had children before they know that they're going to have it. Anyway, it's a progressive dementia and chorea, which is the uncontrollable movements of the limb. Again, it was first described in the late 1800s by George Huntington. And it can be a problem. I mean, you know, you have the children and then you find out you have this, and then the kids have to wonder whether they have it. And then there's the whole question of whether you're going to get tested for to see if you have the gene or not. Other disorders with this, of course, include Alzheimer's, hemochromatosis, and obviously breast cancer. So far, we've looked at mosaics, disorders that have a reduced penetrance or an age-dependent penetrance, and now we can have disorders that have a variable expression. Now we're talking about the severity of the disease, and that can vary greatly as well. Penetrance and expression, you have to be aware, are distinct. Penetrance, one has the phenotype or not. Expression refers to how much of the disease that you have. Now, what can this variability be due to? Environmental effects. Do you have a good diet? Do you exercise, or do something stupid like smoke? And then you have modifier genes, other genes that can affect the expression of this. So, or even worse, you can have what they call allelic heterogeneity. This is when somebody, again, it's sort of like a digenic situation. They have the sickle cell on one chromosome and beta thalassemia due to different mutations on the other chromosome. So that can make the expression more severe and, of course, variable. Another thing that can happen is something called locus heterogeneity. This is when you're talking about a single disease, but it can be due to different mutations in entirely different genes. So they have the same phenotype, but they have different genotypes. And we see this in different families with the same disorder. An classic example of this is adult polycystic kidney disease. It's an autosomal dominant disease that leads to the formation of all these cysts in the kidney, and then they go into renal failure. And so the patients have cysts in the liver. Sometimes they have hypertension. Well, of course, that makes sense with the liver. And then cardiac defects, and sometimes even aneurysm. And it can be due to the result of two mutations on not entirely different genes, but they're on different chromosomes. So they're called PK, polycystic kidney disease 1 and 2. But they have similar functions. They're what we call membrane glycoproteins. So they're in the cytoplasmic membrane of a cell, and when they bind, they activate the cell, and usually they initiate the cell growth. So you have too much cells growing in the kidney, forming these cysts, and this is not compatible with a fully functional kidney, so they can go into renal failure, as we'll see on the next page. And this shows you just how severe this can be. You can see all these multiple cysts in here are a result of excessive proliferation. So the mutation in this case was a gain in function where the protein was always on, and so there were just way too much proliferation within the kidney. Well, you're thinking to yourself, when is this going to end? We have so many different things that can happen to the disease transmission. Well, it's not going to end just yet. Anyway, pleiotropy is another thing that can kind of get you confused as to what's really happening with the family. And pleiotropic genes are simply genes that can have multiple effects on the human body. So they're located in maybe separate organs, and so you'll see different types of responses in different people, but it's still the same gene. Marfan syndrome is a classic example of this. Again, an autosomal dominant disorder, mutations in the gene that they call fibrillin. And so this affects the skeletal system, and so you can see some really nasty little bowing. But another one of this is like cystic fibrosis, because that's a very large gene and they have multiple mutations in that one. And so, but it also will affect other endocrine organs as well, albinism, and then what you're seeing over here is some examples of osteogenesis imperfecta. And you can see how severe it can get as you go further to the right. Genomic imprinting is a fairly recent discovery, maybe 15, 20 years ago. When Mendel did his work with the peas, he showed that a particular phenotype, it didn't matter whether it was inherited from the mother or the father plant, if you will. But that's not the case with humans. Some genes are only expressed from the maternal or paternal copy. So if you lose the maternal copy, okay, and you only have the paternal, then you have lost both genes in a sense. So the classic example of this, well, classic now, are two different disorders on chromosome 15, and depending on whether you inherit the it's actually a fairly large deletion for mom or dad, you have these two different disorders. So it's a deletion of 3 to 4 megabases. So we're talking about 3 to 4 million base pairs. So that's a fair amount. And if you inherit this deletion from your dad, you have Prader-Willi. If it's from mom, it's Angelman. And it's just because, depending on whether you inherited the chromosome 15 from your mom, you have a certain set of genes that are active and another set that are inactive, and it's reversed with your father. So if you lose one region, say you lose it from your mom, you've lost those things that are inactive, that should be active on your mom, but it's inactive on your dad. So you have these different disorders. Angelman syndrome, severe mental retardation. They have seizures and problems walking. The lack of production of a protein involved in recycling of proteins in the cell. And this is particularly important during brain development. In the brain tissue, this gene is only active on chromosome 15 inherited from the mother. So if the mother has the deletion, the protein is not produced. On the other hand, Prader-Willi syndrome, they're fairly short, hypotonia, so small hands and feet, have a tendency to be obese. They're hungry all the time. And they'll have anywhere from mild to moderate mental retardation and hypogonadism. Again, several genes are involved, but in this case, they're expressed in the brain in the father. So if the father has deletion, now you have Prader-Willi. And this is an example of the two. The Prader-Willi, you can see the weight problem, and the Angelman, they used to be called the happy puppet disease because they looked like they were always being manipulated by wires and this sort of thing, but they try not to use that term anymore. So how is it that on the mother, some genes are made and others are inactive, and it's the reverse in the father? It's the same chromosome. So what causes this difference in pattern of expression of these genes? And many aspects of this, of course, are still under investigation. But we do know that in some cases, the amount of methyl groups that are added to the bases in the DNA, this will lead to the inactivation of that gene. In other words, the RNA polymerase can't go in there and make messenger RNA, and without messenger RNA, you can't make the protein. So the DNA methylation will inactivate transcription, and methylation patterns vary between males and females. We also know that histones, remember histones are proteins that have DNA wound around them, sort of like the DNA being the thread on a spool. The histones are the spool. If the histones have very few acetyl groups on them, they bind the DNA more strongly, and that prevents the RNA polymerase, the enzyme, to get to promoter regions and start transcribing a gene, or in other words, converting into messenger RNA. So it prevents the unwinding. So this is another way that you can shut down a certain genes. So we figure altered chromatin structure probably includes both methylation and the histone hypoacetylation. So there are very few acetyl groups. And then, of course, the patterns of supercoiling would also affect gene expression. So we're back to gene expression again. Well, there's yet another disorder known as anticipation and repeat expansion. This is a situation where you've got a disease that seems to get worse as you go on with the generations, and the most recent generation being more severe than the previous generations. So this is referred to as anticipation. Well, what's going on? Anticipation's really just meaning that there's an increase in the number of these little triplet repeats that are present in a gene, usually before the gene. And many of these genes have these simple sequence repeats. In fact, in some genes, if the repeats become too numerous, it can inactivate the gene or give it an abnormal function. classic example of this, I know I've saying classic example for all of these, but these are the ones that occur most frequently, so that's why it's always the classic example. But anyway, it's called myotonic dystrophy. This isn't something you want. It's an autosomal dominant disease. It's progressive, has skeletal muscle deterioration, insulin resistance, cardiac arrhythmias, testicular atrophy, and cataracts. Okay. There's an affected gene. It's a protein kinase on chromosome 19. And protein kinases, again, are very important in internal cell signaling, which leads to a change in gene expression. So we're back to the central dogma. But anyway, the mutation is related to the number of these repeats, CTG. And these are found downstream, I should say, in this case, of the untranslated portion of the gene. If the patient's normal, they have anywhere between 5 and 37 copies of it. If they're unaffected or maybe mildly affected, so they have, kind of a middling number of repeats, 50 to 100. Severe, they have 100 up to 1,000 copies, and the number of repeats often increase with succeeding generations. So here's pictures. We've got 3 generations, the grandmother, the mother, and the child. And you can see evidence, particularly the shape of the eyes in each of them. You can see how much more severe it is on the younger generation. So you can see that this, here we've got the A, B, and C. This is a blot showing you the increase in number of repeats. Why does it go up here? It's because as you gain more repeats, the fragment gets longer, and so it doesn't move as well on a gel. So this is why you're seeing it this way. And here, what you're seeing is with the pedigree up here, is that you're seeing that, the original father who had it was 55 years, and then in this next generation is between 48 and 50 years that expressed, and here again in this third generation, now it's between 41 and 40 years. So how does a mutation, in this case in the 3' untranslated region, so it's at the end, past all the exons and everything, cause so many symptoms? Well, it turns out that this mutation leads to the formation of a messenger RNA transcript. It remains in the nucleus, and it produces a sort of gain of function activity, as it says there. So this abnormal messenger RNA interacts with RNA binding proteins that regulate RNA splicing. So what happens is you get this alternative splicing of several different proteins, and this causes all the different symptoms. Another repeat expansion, CTG, alters genes on chromosome 3, and this also leads to a similar disorder. In fact, they just simply call it the same name, myotonic dystrophy, because it's the same constellation of symptoms. This leads to a discussion of Fragile X. In the 1800s, they noticed that males more than females were institutionalized for mental retardation. And we now know that this is partly due to something called Fragile X syndrome. And it counts up to 40% of all cases of X-linked mental retardation. Now, most inherited causes of mental retardation are Down syndrome, which, of course, these are sporadic. They're not necessarily inherited from one generation to the next. But Fragile X, like Down, has a distinctive facial appearance. They got a longer face, large ears, very hypermobile joints, and macroorchidism. Anyway, the mental retardation is milder than with the Down syndrome and less common in women, and that's because it's on the X chromosome, and women's X chromosomes women are all a mosaic because the inactivation of the extra X chromosome is random. So it will have a tendency to be milder in women. And this is just showing you boys with Fragile X syndrome, and you can see the very classic features, the long face and the big ears in them, and of course they all have a pretty cute smile on them as well. Fragile X is another repeat expansion type disorder. In this case, it's in the 5' before the start of the gene, and this gene is known as FMR1. And it's a gene that's made in neurons. And in this case, the repeat is not CTG, but CGG. Anyway, normal, you have anywhere from 6 to 50 copies. If you have an intermediate number of copies, 50 to 230, this is what you'll see in normal transmitting males. They'll transmit it to their female offspring. Those who have Fragile X syndrome have anywhere from 200 to 1,000 copies. And it turns out that males with intermediate numbers do not have affected daughters because for some reason there's no expansion of the repeat. But females with an intermediate copy can have repeat expansion and transmit it to the offspring. So we see expansions with the daughters, with the females, but not the males. What is the function of the FMR1 gene? Well, it might not surprise you to find out that the highest rate of transcription in the human body occurs in neurons. In other words, there's more messenger RNA in a neuron than any other cell type, and that only makes sense if you think about all the functions of neurons. Well, it turns out the protein product of the FMR1 gene binds to messenger RNA molecules and helps to transport them from the nucleus to the cytoplasm, where they can then associate with a ribosome and have the protein made. And also in the cytoplasm, this protein may also help to regulate messenger RNA translation. So it could bind to the ribosome or do something like that. We're still working on it. But down below here you actually see a sort of what we call a ribbon and space-filling model of the FRM1 protein. And here you just kind of found this online. You see that if you have the normal number of repeats, you have the messenger RNAs and you get the protein made and it can do its job. If you have the intermediate number between 55 and 200, now you get excess messenger RNAs made, but a few less proteins. Now, if you have the full mutation, though, greater than 200, now you don't get as much messenger RNA, and you have none of the protein being produced, and you have Fragile X syndrome. And since this is all occurring in the neurons, you can see why it has such an effect on cognition. Well, you'll be happy to know the final inheritance pattern that we're going to talk about is mitochondrial inheritance. Now, there are some rare diseases, but they are being recognized more often these days because we are more aware of them than we used to be. And they're results of mutations in the mitochondrial DNA. Mitochondria have their own DNA or genome. And it also has a very characteristic mode of inheritance. You're going to love it when you see the pedigree on this one. And but it also has a very large degree of variability or disease expression. So if you think about it, mitochondria DNA, everybody knows mitochondria are the powerhouse of the cell, so they make all the ATP. This is the site of energy production. So these genes, if you will, the genome, in the mitochondria, you have several copies of it. It's fairly small. It's actually very similar to a bacterial chromosome, if you want to know the truth. And it encodes proteins that are important in generating ATP, oxidative phosphorylation proteins that are important in the electron transport system. So a replication and transcription occurs in the mitochondria independent of the nucleus. So the mitochondria are definitely different. But because you have so many different mitochondria in a given cell, you should have quite a few copies. And depending on the cell, you may have 100 copies or more. And not every one of the mitochondria will have the disorder. You can automatically see that it can be very much kind of a mosaic type inheritance pattern. So this is just a copy, or I should say a picture, of the circular mitochondrial DNA genome. Unlike our chromosomes in the nucleus, it's circular as opposed to linear DNA. And all these numbers and everything are just telling you some of the genes that are present in this DNA. Don't worry about knowing that. Now, it turns out that mitochondria DNA has a very high mutation rate because within the mitochondria there's no DNA repair mechanisms. The other reason that it is kind of easy to figure out the inheritance is that mitochondria are transmitted only through the maternal line. So mother transmits the traits to all of her children, male and female. Nothing is ever transmitted by the males. So since each cell, as I was saying in the previous slide, contains many mitochondria, some of which will have the mutation and some don't. So we distinguish it from mosaicism by referring it to as heteroplasmy. And that's the cause of the variable disease expression. So the unequal distribution of mutant mitochondria upon cell division means that in some eggs we'll have more mutant mitochondria than others, and the somatic cells have some organs affected more than others as well. Now, typically, mitochondrial disorders seem to affect organ systems with high ATP requirements. Not too surprising. So once again, it's not surprising that we see the disorders in the central nervous system. So these are just some of them, and as you can see, the timing with this got a little out of whack, so I've just brought everything up. One of the more common mitochondrial diseases is known as Leber hereditary optic neuropathy. Sometimes when you're going over and over and all these sorts of things, sometimes your mouth seems to go crazy, or at least mine does. Anyway, these unfortunate people have a rapid loss of vision, again, as a result of death of the optic nerve. Begins in their 30s. It's usually irreversible, and the heteroplasmy is rare, so it has a tendency to have more of the mitochondria affected. Then we've got something called myoclonic epilepsy with ragged red fiber syndrome, MRF. I actually had a graduate nursing student who was diagnosed with this, although hers was not as severe of this. So she would have problems, though, with seizures. Dementia, ataxia, myopathy. I think she had some of the myopathy, but she did well in the class, so certainly she was not demented. Again, this can be heteroplasmic, so it's highly variable. And look at this. This is a mutation in a tRNA gene. Now, think about that. You've never heard of mutation in tRNA genes in the nucleus, and that's because that's not compatible with life. Because that would mean that the majority of your proteins wouldn't have the right amino acid sequence because the tRNA wasn't reading things right, and you can't survive under those circumstances. But in the mitochondria, which codes so much fewer genes, you can, especially if it's not all of them have the defect. Anyway, involvement in some, it turns out that some mutations in mitochondria do affect other more common human diseases. This is some forms of deafness, and then there's the mitochondrial encephalopathy and stroke-like episodes, and we think it contributes to some cases of Alzheimer's. So this is what I'm talking about, the real nice pattern. You can see the inheritance pattern without any problems. We start off with the original affected female, and of course she will have affected offspring, but the male will not be able to transmit it, only the female lines and the female offsprings. So that's it for chapter 3. We'll move on to chapter 4, and I hope you have a lovely evening or morning depending on when your shift was. More later.